I'd like to now call up Juan Van Doorslaer, who's in Alison McBride's lab at NIAID. He's going to give us a 10-minute presentation about software tools that are now available to analyze HPV genomes or PV genomes that are coming online in the tidal wave.
Good morning. I have no conflicts to declare. I don't have a slide, but I didn't take money from anybody. So good morning, and I want to thank the committee for inviting me to give a talk this early in the meeting. And I'm hoping that this will give us a lot of time to interact, because we really want to get suggestions and ideas from all of you on how to improve PAVE to make it a tool that will be helpful for the entire community. I think a great time for this type of interaction would be tomorrow when we're hosting a breakfast session in room 209. And we're going to outline a little bit what we want to do with PAVE, what we think the next steps of PAVE are going to be. But we really hope that you guys will come out and give us ideas on how we can make PAVE useful for everybody. So hopefully most of you have seen the homepage of PAVE by now. For people that have not, I've indicated the homepage, the address. In addition, it's probably easier to just Google PAVE NIAID, and it should be the top hit that comes up. So the problem has been for a long time that there has been no central resource for papillomavirus sequences, basically since the Los Alamos National Laboratory Database ceased existing in the late 90s. The problem with NCBI and EMBL and databases like that are that there are hundreds of thousands of sequences, PV-related sequences on these databases, but they're not really annotated well. Lots of them contain errors, so it's really hard, unless you really know what you're looking for, to find all the sequences of interest. So a major goal of PAVE has been to host uniformly annotated reference genomes for each papillomavirus. And the idea is that people with different interests, epidemiologists, clinical scientists, molecular biologists, evolutionary biologists, that we can talk, that we know that when we're talking about a papillomavirus, be it HPV 16, HPV 45, that we know that we're talking about the same virus and that we can communicate changes in these genomes to one another. So PAVE is an acronym for the Papillomavirus Episteme, and episteme is etymologically derived from the Greek word for knowledge or science. And Plato defined an episteme as the body of ideas that determine the knowledge that is intellectually certain at any particular time. And I think that really reflects well what we're trying to do with PAVE. With PAVE, we're trying to give you guys sequences that we believe are true. We're not trying to provide you guys with hypotheses. The idea is that you take the sequences, run them through your analysis, look at them, study them, and come up with hypotheses or use these sequences to test your hypotheses. So as of July of 2012, we have 241 completely annotated viral types on PAVE. You can browse and use 2004 PV proteins and 2,245 genes and regions. In addition to sequence data, we're also providing 48 PV proteins for which the structure has been solved. We've starting to trying to annotate splice sites, and one of the things that we're showing at this point is the E1E4 splice. In addition, what we've also done is we have re-annotated the L1 protein so that all the L1 proteins in PAVE now start at the same position based on a conserved upstream splice site. So hopefully that will again help the community. And at the bottom, I'm just showing a phylogenetic tree of the 241 viruses that are currently in PAVE. So the way that most people probably approach PAVE is through the search engine. You can easily use our search engine, and I hope you can see it on the screen. You can start off with keywords. You could look for HPV 16 or HPV 31, for example. But what's really cool about the PAVE search engine is that you can use a lot of really powerful filters to really search and get down to the sequences that you're interested in. And in a couple of mouse clicks, you can download whatever sequence you want onto your own machine and do your own analysis. But in addition to just being a database, PAVE is more and more trying to develop tools to make analysis on the website possible. So one of these tools is what we call the locust viewer. And in this locust viewer, I'm just giving a linear representation of the HPV 16 genome. And on the top, in the yellow bars, we have shown all the annotated open reading frames. In blue, we're showing the upstream regulatory region or the promoter region, and we're indicating where all the E2 binding sites within that region are located. In addition, like I mentioned, we are now showing the E1, E4 spliced protein. So if, for example, you click on those red bars, you will get the spliced protein in the bottom in the selected feature window. And you can then automatically click on BLAST to look for more E1, E4 proteins in the database. And you also get on the bottom the nucleotide sequence corresponding. So like I mentioned, one of the main goals of PAVE was to re-annotate and to get a uniformly annotation for all the known papillomaviruses. So the way we did that is we went into GenBank and downloaded the sequence for all known papillomavirus types. I then went in and isolated the individual open reading frames and used the BLAST-based approach against a custom database to annotate all those proteins. So this is all done computationally. At that point, I move into quite an extensive manual control phase, where the first question I ask is, are all the features that we're expecting to be there, are they all present? So I'm looking for, is there an E1 and E2 protein? Is there an L1 protein, L2 protein? And in most cases, you're expecting an E6 and E7 protein. If everything looks okay, I then make multiple sequence alignments of these annotated proteins. And again, look at these alignments to see are the lengths of the sequences, do they make sense? Is there anything weird? Does it look like there was a frame shift that occurred? And if everything looks fine, at that point, the sequences are added into the database. However, if at any of these manual control steps something looks wrong, like there is a stop codon in E2, for example, I try to go in and manually look for what the problem is and see whether I can edit that sequencing error, that mistake, and at that point, only at that point does that sequence get placed into PAVE. So since PAVE really is a bioinformatics tool, what we want to provide users are sequences in which all the expected features are present. And if an expected feature is missing, what we're trying to do then is use the maximum amount of evidence to use the minimal amount of edits to fix those mistakes and to reintroduce that feature that we are expecting. So the sequences, in those instances, the sequences that we have on PAVE will be slightly different from the ones at NCBI, but the idea is that these sequences are going to be used for bioinformatics approaches as a reference, and we are calling them in silico genomes to amplify the idea that these are not necessarily real clinical isolates. So for example, imagine a virus in which the E2 protein would be disrupted because of a stop codon or a frame shift. I will then go into GenBank and PAVE and look for all the variants or closely related viral types that we can find to this particular genome, align the sequences, and then look at these alignments whether I can find the reason why there would be a problem. And I think if you look at this alignment, I think you can appreciate that there's, in the fourth virus from the top, there's an A missing. And just reintroducing that A in this whole stretch of repeated nucleotides reconstituted the E2 protein in this virus. So like I said, I think this is really as a complementary approach to what the NCBI is doing. All the sequences on NCBI are maintained and are not changed, whereas the sequences on PAVE are more of a living tool that where we're trying to provide users with edited viruses that have all the features that we expect there to be. Like I mentioned, in addition to just sequence information, we have 48 proteins for which the structure has been solved. So if you're interested in a structure, you can just go to what we call the reference PDB file, and you can just click on one of the viruses of interest. So for example, you're interested in L1. I'm showing here the structure of 16 L1. And this window is just like any other structure viewer. You can rotate, manipulate the protein however way you want it. You can color it. But what's really neat about the PAVE structure viewer is that right below the gray window, there's a pairwise sequence alignment between the structure of interest and a protein of interest. So let's say you want to compare HV18L1 to the HV16L1 structure. All you need to do is in the window on the right side, select HV18L1, and what PAVE will automatically calculate is a pairwise sequence alignment between HV18L1 and HV16L1 structure, and it will highlight those differences in blue. You can then go in and select the changes that you're interested in, and they will be automatically highlighted on the structure, so in case you want to make a mutation, you can easily make that mutation. So I'm running out of time, and I don't have time to go into all the other features, but we have BLAST-based searches, we host transcript maps of a lot of viruses, we have a complete copy of the original Los Alamos database, so if you want to find any of that data, that is on PAVE. We have links to variant genomes. We have really cool virus and other tools. We are building a PV classification tool that will be used in PAVE soon. And we're really making a strong push to collect images of confirmed PV lesions. So we're starting to build quite a library that we want to host, but we're still looking for a lot of lesions caused by papillomaviruses. So if anybody has a picture that they're willing to share with the community, please contact us and we can put those on PAVE. So coming soon, we're going to incorporate the variant genomes, we're going to have alignments and phylogenetic trees, and we have an agreement with the Virology Journal to write expert chapters on individual operating frames. So hopefully we'll see you all tomorrow, and everybody has bright ideas on how to improve PAVE for the community. Thank you.
Great job.